why it's showing that. Okay, looks like we're in good shape now. Thank you very much for inviting me here today. It's my first time to Cambridge, and happy to see a lot of familiar and friendly faces who I already know, so good to see friends again, and also to make some new friends here at this meeting. I want to kind of start talking first, what is a digital twin? Because we're going to see this word a whole lot in the next few days. We already have. It's a lot of hype. People try to find a way to put the word digital twin in just about any grant or abstract. And so, you know, the National Academy of Sciences in the US seems to have kind of three elements that they say is a digital twin. First of all, it needs to be predictive. There's some kind of a model that can say, given your state now, can you make a prediction for the future? It needs to be twinned to an individual. Sounds obvious, but you'd be amazed what people try to get away with. And also, it's not just a one-time calibration. It's something that is continuously updated over time as you get more patient or individual information. So what is not a digital twin? Most of the things that I build are not a digital twin right now because we can calibrate to an individual patient, but we're not updating them in time. We're missing that data assimilation step. A lot of people build stratification models. Those are groups of people, not really individuals, even though you can evaluate that model for an individual, so not really a digital twin. Virtual trials are fantastic, and we're in love with them, but they're populations, generally speaking, although sometimes they're based upon calibrations to individuals, like Sandy's really beautiful work this morning. Most cancer atlas projects are, you know, built upon a consensus model of many imaged patients, and also doesn't make it much of a twin if you destroy the samples when you build your model. The thing you twinned is gone. GE is not going to destroy the jet engine when they build a digital twin. It's not very useful at that point. High-resolution patient imaging, right? It may or may not be updated or may not be predictive. So there are a lot of things that are not digital twins, but there are a lot of things that are the components of a future digital twin. So I think in this group, we probably have almost all the pieces we need, or many of them, to combine them together to create a future patient digital twin. And I just want to make sure that I don't claim that I've done it, because it's really hypey. We need a lot of things to create digital twins. We need, first of all, if you're going to build a digital twin that's calibrated to a patient, you better have a class of models that are worth calibrating. Things that can span the expected ranges of behavior. So you can say some patient is going to have probably some parameter values in this class of models. Otherwise, what's the point of doing the rest of the work? Then you need to be able to identify how much data is it going to take to calibrate this kind of model and what you can do with it. You need good and robust and practical methods to actually calibrate it. And then having built the model, that element number 3, you need a way to assimilate data to continuously update this model. And then given a model, then you need to be able to do the beautiful things like you saw in the last talk to explore it in high throughput, to say, can I interrogate this model and identify some things to do for that patient, right? And you need to be able to kind of do all this. So we need all of these pieces. In this talk, I'm going to talk a little bit more about how to build models themselves and a little bit about how to say, if you have a model, how do you export in high throughput to get something out of it. My lab really doesn't do this very well, and we're looking for the right people who do. So I want to make very clear about what we do and don't do in our lab. So, you know, for me as a mathematician, you know, it was very eye-opening. I started off as a continuum guy, moving boundary problems, and then looked under a microscope with a breast pathologist for the first time and said, this isn't going to work. There's just the individual cell effects, watching clusters of immune cells nearby the tumors in ductal carcinoma in situ, saying, holy moly, there's some kind of a big dialogue between all these different cell types, and we're nowhere near equipped to describe them with our simplistic ODE and PDE models that we were doing at the time. And so I really took a step back and saying, it's really interesting to say that at the molecular biology level, it's just mind-boggling, baffling, redundant, complicated. I have no idea what's going on there. I can't get my head around that chemistry. But if you go one step up, you say, what are cells doing, right? That there's a handful of things they do. They can divide, they can die, they can push each other around, they can eat each other and attack each other. It gets really fun, right? So individual cell behaviors, pairwise interactions like sticking to each other, pushing each other around, performing random walks, again, eating each other, it's really fun. I love macrophages. It's like Helen and I had a great talk on the way up here on macrophages because they're just so fun. Cells attacking one another and, you know, and other forms of contact communication, then put this in an environment with constraints, things like diffusion limits and mechanical limits. And from this small, relatively small and finite palette of things that individuals do in interactions, you put it up and scale up over thousands or millions of cells interacting, and all of a sudden you have interesting, complex ecosystems. And so, you know, systems biology, in particular multicellular systems biology, says, can we tease out the interactions and the individual behaviors and scale them up to predict what's going to happen in this complex system? And then can we go backwards and do engineering and say, this is a complex system as it is today. I'd like to steer it to a different state, such as non-cancerous. Can I choose interactions and interventions to say, tune down this behavior, turn up this interaction, add this new cell type, identify what those interactions ought to be, so that we can steer the system in the direction we want to go. And that's basically clinical intervention. And so we need a conceptual framing to help us tease out the system and build models. And if we can build models that are descriptive and predictive enough, now we can start tuning them to choose those interventions. So we think a lot about these cell-cell interactions as kind of a signal response framing, kind of like a cell communication ideal, that some cell will release a signal, and that signal is not necessarily a secreted chemical factor. It can be physical signals too, like pushing on each other, contact, right? So all sorts of signals, but think of it as the input to an interaction. And then there's a receiver who can listen in and find that signal, and then they have to have a behavioral response in response to that signal. So an example would be a macrophage, a sender, secretes a chemical factor, IL-6, that's your signal, which reaches another cell, CD8 T cell. So there's a recipient who responds to that signal by changing, say, their chemotactic behavior. So a sender, a signal, a recipient, and a response. If you can kind of tease those out and separate them, say who sends signals, who receives signals and responds to them, then we can start teasing out this system and build a dynamical model. So we use agent-based models, which is not going to be a surprise to anyone who knows me, to build these kinds of systems. But the nice thing about an agent-based model is that it models the individual cells, they have their individual states, and the individual processes that act upon those states. And so if we can kind of dial it back to that palette of individual behaviors and implement them one by one, then we can worry about how the signals change those behaviors as model rules. And so we usually then say, now they have those agents, those objects that have properties and rules that act upon them. I can place them in a virtual environment and model, say, the diffusion of all these different chemical signals that are in the environment, and do lovely things like random walks and chemotaxis. And so in that framing, our cell agents are signal processors. This agent in this space, at this point in time, in this particular location, can sample a variety of signals. I can't sample something that's far away from me. I have some kind of a range of vision, right? But I can sample a handful of signals to say, here are some chemical factors, here are the gradients, here are the cell types that I'm in contact with, here's my mechanical pressure. So not all signals are chemical, right? They could be mechanical. And then I can decide what to do with those signals. I process those in some way to say, now I change my proliferation, change my migration, change my secretion, change my phagocytosis and my T cell killing, whatever it might be. And so if we can really think of those become the rules and our agents. So we've been kind of using that to form a virtual laboratory to study these kinds of questions. And I think all of us have really been forming our own virtual laboratories to study these questions. And so we started off saying, several years ago, we knew we had to solve a whole bunch of reaction diffusion equations. And that's always, sadly, the most expensive part of the computational problem. So we solved this part first by saying, let's make it a finite volume method. I'm going to turn it into a big vector of very similar forms of diffusion equations. We could just vectorize the heck out of it. And then we just did, x, y, z, over and over and over again. But because it was all nicely vectorized, we were able to get an efficiency that says that as you increase the number of diffusion equations, you don't double, triple, quadruple, quintuple the computational effort, that slope is less than 1. So simulating 5 or 10 or even a dozen diffusion equations, even on a desktop, is not too bad. And so that's quite nice. And in particular, we built into that there are point sources and sinks so that cells can secrete and sense things. But right now, there's just dumb sources and sinks. So on top of that then, we added an agent-based framework on that to say that the sources and sinks are now cells, not just dumb static things in space. And so we added in the individual behaviors and the variables and then started kind of tracking that around. We made sure that this is cross-platform, so it runs equally Linux, Windows, wherever you might want to run it, without having to change your model or your configuration. So we published this several years ago, and this is kind of the basis of our computational lab. But there are a lot of challenges for building agent-based models kind of at that fundamental low level. Not sure what that is. First of all, coding and implementation. You have to have some, a lot of software knowledge to write these things. You have to understand a really huge user's manual of an API to access things. How do I get this signal? What's the way to sample the gradient of this location? Do I have to calculate? Do I have to hand code some kind of a center difference? And then debug that. And then you have to go into the API and say, how do I actually change this behavior in this cell type and write it and avoid segmentation faults and do race conditions, or hopefully not do race conditions, and lots of debugging, right? And that is then, once you kind of come up with a model structure, then you have to figure out how to update it, how to add new rules, check it against data. There's a lot going on here. But some of the challenges really are just because of the static model structure, because it takes so long to build the thing in the first place. We kind of fall in love with our model, and it took so much work to build the thing that we don't want to change it. And we don't want to add a new rule or remove a rule, because my goodness, I just spent 6 months debugging this thing. So the difficulty of building new rules and modifying them and checking them and being willing to throw them away, I think, is an impediment to the way we do modeling. So there are some advances to address some of these challenges. For the exploration, high-performance computing, which we'll talk about for just a moment. And really, what we call HPC is more like HTC, high-throughput computing. We're not really worried about making one humongous simulation really fast. I want to run 100,000 simulations pretty fast. That's what I'm more interested in my class of problems. And then we'll talk a little bit about making it easier to build these models. So I want to kind of go around an illustrative project, and this is with, so this, I probably should have not put the name under here. This is Ilya Shmulevich. Unfortunately, we lost him last year to cancer. And so we did this project together for several years as he was winding down. Miss him. And just a brilliant friend and scientist, so please, if nothing else, give us a look to look at his former work. And then led by my postdoc, Aber Russia. And so really, we were very interested in saying, could we build a class of models to model micrometastases arriving in the lung, early immune interactions, and saying, there are a lot of parameters in this thing. How on earth are we ever going to build a digital twin? Because we have too many parameters to calibrate. And so we decided to take a step back and do a little bit of analysis of this model. First of all, really asking two questions. Is this prototype tumor immune model that we built sufficiently expressive that I can see the range of expected patient behaviors, right? If it doesn't span the space of possible trajectories, it's not worth building as a digital twin because it doesn't capture all the patients I want to capture. And then if it can span that, we kind of start analyzing in high throughput the possible trajectories of that class of models and turn that high dimensional space into a lower dimensional space. Can I partition parameter space into kind of regions where in this region the trajectories are similar? And can I make broad classes? Our hope, our hypothesis at the time, was that we could divide parameter space or patient space into templates, a lower dimensional set of saying 13 or 12 possible template, trajectories, and say, could I take, say, the centroid of one of those patient classes as my starting guess for that patient, and then assimilate data over time to refine that model of that individual. And then I'd be getting towards those three elements I needed for a patient digital twin. Something mechanistic and predictive, so they can calibrate to an individual and assimilate data over time to improve the model and the predictions. So can we start off by saying reduce the search space to something much more finite before we move on to data assimilation? So the model we took actually started off in COVID-19 as a completely different model. It's a model of infected tissue with immune cells coming in and responding to it and kind of clearing out. So we had a viral dynamics model to get the whole immune compartment. And the key part of that model actually was that it was a local model, the spatial model of agents coming in and clearing out infected tissue and everything, and also a non-local model, a system of ODEs of the distant lymphatic system where immune cells would leave, go and ramp up development of CD8, like effector cells, and then come back in and start attacking the tumor. So kind of that two things. So we stripped out the virus and then added in micrometastasis and then kind of adapted that model. It's just a basic starting point. And so our approach was to create a large cohort of virtual patients, so very similar to the beautiful work we saw this morning of drawing random values in a large high-dimensional parameter space and creating virtual patient parameter sets, so just kind of sampling randomly for now. And then remembering that it's a stochastic model, for each individual patient, a parameter point in space, run multiple replicates to see what the trajectory would be for that virtual patient, because we don't really know what's going on, right? And then to say, can we partition that input space based upon those simulated trajectories to come out with those patient templates? So, first answer is, yes, the model is expressive. If you vary over the parameter space, we can get the kinds of behaviors we would expect in a class of patients. Some tumors could be successfully resolved and eliminated by the immune system. some would grow completely away, and some would be maintaining some kind of a partial marginal control. So we got the kind of class of behaviors we expect. So, okay, this is a model that might be worth trying to calibrate to individuals. Answer number 2, unfortunately, is no. We found that for many individual parameter values, and again, this is like a thought experiment saying, suppose I knew with infinite precision the exact parameter values for an individual, can I predict what happens in the future? And even with that exact initial condition, exact parameter values, there's humongous divergence in the trajectories. And so here's like one example of a simulated patient. So you can see that at long simulation times, we have some tumors that are completely eliminated, some that grew completely away. And say, okay, well, that's a huge divergence in the trajectories. And I trace it back, it's like, oh, that's about where things looked about the same. But then you dig in. So I love how the previous talk, right? You said you can dig into the models to get an explanation of what you've seen. So here we dig in, right? And you say, we've seen that actually it's a little bit farther back where the macroscopic scale still looks deterministic, where there's a very good agreement against all replicates. But if you go a little bit deeper in, it's the early chance encounters between the immune cells. Tumor seeds in some location, and sometimes by random walking, the immune cells find that tumor just early enough to go retreat over to the lymphatic system, build up the cavalry, and the cavalry comes into the rescue, control the tumor. But if that stochastic interaction happens just a little bit later, it's really tough to catch up with exponential growth. And that was it. So, but that tells us that, for these kinds of models where we really care about stochastic interactions like tumor immune models, it's probably not going to cut it to do like phase portrait analysis to look at the classes of patients. So we definitely need much more sophisticated data assimilation to do this kind of class of problem. But having a model that we could explore a whole lot in advance allowed us to answer some questions very early on without going through the expense of trying to run a virtual trial and do the kind of calibration effort and then have very little report and grant progress reports if we were lucky to get a grant at all. So sometimes a no finding is a good finding. So those are the kind of insights. I basically just said these, so I probably should just move on from it. So we can build models, we can explore them in high throughput computing, build lots of parameter sets, run thousands of simulations at the same time, which is really great because you can ask certain classes of questions that you can't ask if you're just running on a desktop. But the problem still remains that models are hard to build, right? Lots of manual curation, lots of manual learning, hand coding of these things. So we want to simplify that process. So, kind of going back to saying, how do I build a model, right? We have to formulate your hypotheses to say, what kind of cell types and signaling factors do I think are important in this particular problem? And sometimes we talk to domain experts, sometimes we learn it from the literature. Hopefully in the future, AI can kind of tease these things out, saying, hey, Siri, what's important in breast cancer? And it probably tells me something wrong or says, you could do it in the app. But maybe Gemini will do something for us. Can we identify the biophysical signals that we think are driving the behavior? Can we start writing out these hypotheses, as actual words, say, doxorubicin increases apoptosis, or some signaling factor changes transformation of macrophages. It requires a real dialogue between biologists and clinicians who have the domain expertise. And we, in this room, have the domain expertise in writing models, and you can't do it either side alone. Then you have to turn those hypotheses into mathematics, and this is something where we kind of go off and do our thing for a while, and then we have to code it into actual code. Right now, for most of us, this is artisanal, handcrafted C++. It comes with artisanal bugs, and takes a lot of time to debug. You debug, finally runs. Come back in a few months, talk to the biologist, and say, hey, I've got a model, let's take a look at this thing. And they said, I'm sorry, but I don't remember anything we talked about 3 months ago. And it's really hard to make that iterative progress with a friend. And then, of course, handwritten models have a lot of problems, especially if you're writing them as a manual logic. Where you insert the rule in your network of rules makes a big difference, right? And so you need to be really careful of that as you add more rules to your model, you're not breaking your model and your logic as you go. So these kinds of if-then flowcharty rules were really popular in cellular automaton models and things like this, 10, 15, 20 years ago, but they don't scale very big before you start seriously breaking them by adding new rules. And then, of course, the more complex the model gets, I found it kind of has an 1 over n effect, that as the number of rules or lines of code kind of goes up as a function of n, the number of people who can understand that model goes as like 1 over n, And eventually 1 over n is less than 1, and no one understands the model, and the problem breaks, and you just can't go any further. So we really want to avoid that. And in particular, we want it so that not just we in the room can code rules, but biologists and clinicians can have a direct role in contributing to these models. So that's where we want to go. So our goal is to create a computable model grammar. We want it, first of all, to be human readable, that a sentence in our grammar says, you know, some signal changes some behavior, some cell type, that's something that we can read and interpret, say, I understand what this saying is in my model, it has a biological meaning. But also, it needs to be computer and mathematically readable. So you need to be able to translate that word grammar statement into a mathematical response function of some kind, and then translate that into code in real time. And we also want to worry about this complexity issue of where I insert the rules matters. So we're going to make sure that we make this grammar so that order of execution doesn't matter, that new rules don't break old rules, and that's kind of our thing here. So key elements, for us to say, you know, take that step back and say, I need some kind of a dictionary of the signals or the inputs to rules that has a consistent naming that I can access very easily in my framework, in my language. And then I need a library of reference behaviors to say, I have a way of write cell cycle. I have a way that I write death models, and migration models, secretion, migration, uptake, phagocytosis, effector attack. And so I have a library of core cell functions that are implemented with a well-defined mathematical form. So I can go and find like supplemental material, say, okay, this behavior has these 5 different parameter values. I know what they mean, and I can map that parameter to human language symbol. Then I need a grammar that connects them together. I basically need a subject, a verb, and a predicate, right? Some signal increases, decreases some behavior, something like that. And then a one-for-one mapping from that onto mathematics and code, which is actually the easy part. So we started looking through, and really we felt like we just needed to build lots of models. So we spent about 10 years building models and looking at other models. In this room has so many great examples. We finally had enough samples to say, what are the typical signals we see as model inputs? Well, chemical factors, their gradients, number of cell types of different types you're in contact with, mechanical pressure and stress and strain. So we came up with whole classes of symbols we found were very commonly used. We hope would cover a lot of the use cases out there. And once you can find a way to sample it and define it and calculate it, it's very easy to make it something that you can query. And then we worked more on the reference behaviors. And again, it helps to see a wonderful dynamic community that has been building models for 10, 15, 20 years. Say, there are a lot of ways to do things, but there's kind of some convergent evolution in this community where a lot of the cell cycle models look the same. A lot of the apoptosis models look the same. A lot of the migration models look like a biased random walk of some kind. There's some commonalities that we can start to kind of standardize on, find that lowest common denominator. And so we built built-in standardized process models for each of these things, and then had a handful of well-defined symbols that go with it. So as an example, cell migration, biased random migration for us. The parameters are choose a speed, choose a bias between 0 and 1. If my bias is 0, I'm just Brownian. If my bias is 1, I'm deterministic. And to say, now choose a bias direction, say a chemical gradient in this direction. I have speed, and I walk in this direction for persistence time, but I stop, choose another direction, and keep going, right? And you can trace out this random path. And then we added just a little bit more functional form, I guess, concreteness to that, say that for most of our problems, the bias migration, the bias direction is going to be some kind of a sum of chemical gradients, and we'll call the coefficients chemotactic sensitivities. If it's a positive one, you go up the gradient. If it's negative one, you go down the gradient. If it's 0, for most of the terms, you just ignore it altogether. And that was actually enough to express a whole lot of combinations, and this gives us a handful of dictionary symbols that we can put into our models. And so imagine doing this process by process to have a handful of symbols that make sense for those individual processes. So this is why when you write a paper and it's 10 pages, our supplementary material is like 50, right? It's because writing out all these things. So now I have a dictionary of symbols for the input signals, a dictionary of behavioral symbols, and now I just need a grammar, a verb, to tie them together. And we just picked the simplest possible grammar we could to say, in some cell type T, some signal increases or decreases some behavior with optional parameter values at the end. And that's it. And we're like, okay, let's see what we can get away with this simple formulation. But here's some examples. For M0 macrophages, encountering necrotic cell debris, so there's a diffusible substrate, will increase my rate of transformation to an M1 macrophage. It's just a simplistic rule. Well, these kinds of rules, you start putting enough of them together, you start getting something interesting. So now we can write out a rule in a word sentence. Now I just need to do mathematical mapping. So some behavior increases behavior, some signal increases behavior just means I need a monotonic increasing function. We tend to like saturation, so hill functions are lovely for that. And so we just map this onto, you start with your base value in the absence of your rule, and then your rule is increasing it towards a maximal response, and you get the classical response that we're used to. Here's just an example. Oxygen increases cycle entry, that if I'm with no oxygen, I have a very low or zero entry rate, and then I'm going up towards a maximum rate as my oxygen starts saturating with a half max of 21.5 millimeters of partial pressure, kind of going up here, and you get, a response curve that looks about like what we might see. Now, we want to be able to do multiple hypotheses, all integrating to maybe influence one single behavior. So we generalize the Hill response function to multivariate, say that I have all the things that increase my behavior, and I just add them together. So notice that the ordering doesn't matter, right? So adding a new up response doesn't kill my other up responses. And likewise, I collect together all my down responses, everything that down regulates my behavior, and then I take a bilinear interpolation of the up and the down responses to get my ultimate behavioral parameter value. So this integrates up rules and down rules. Adding a new up rule doesn't change the behavior of the other up rules because you can always remove it, and the ordering doesn't matter. So it gets us there. We know simplistic. We know we're not like Turing complete. We're just hoping that we can span a class of interesting problems with this. But it is a rich model space when you think about it. I mean, the dimensionality of the space of models we can make is pretty high. Number of cell types times the number of signals times the number of behavioral parameters times the number of directions it can go. That's already a pretty large space of models. So we think we can get away with a lot here, and we know that we're missing things, and we'll keep adding it iteratively. So it's like language 1.0. But you can also use this to reconcile contradictory rules. So for example, imagine that chemical chemotactic migration. For low density of ligand, you might be have a hard time sensing the gradient. So your bias might be very low. As you get more and more ligand, that gradient becomes a little bit clearer, and so your migration bias might increase. So it's a little bit easier to sense my chemical gradient, so now I'm going a little bit more deterministic. But if you are swimming in ligand and all of your receptors all the way around your periphery are all saturated, you can't detect the gradient anymore. So you expect some kind of a non-monotonic response. And it turns out that you can actually combine two rules, say, C increases migration bias with a low half max, C increases migration bias, decreases migration bias with a high half max, put those together, and you'll actually get that non-monotonic curve that you would expect. So you can actually reconcile contradictory effects with this kind of a language. And because we have a non-moving target, we can start creating support tools. Say, we've been building a modeling tool that says, define your substrates, define your cell types and their base values, and then we have a drop-down system to say, in this cell type, this signal increases or decreases this behavior, add rule. Go and simulate, see what it looks like, and see if you want to tweak your rule, add a rule, remove a rule, and you just do that right there in a web browser. So we're kind of moving right now towards that model. And then once you're happy with your model, then you pull it off the web browser, off the cloud, and you say, go send it off to your cluster and do 100,000 of them. So that's kind of the vision here. So an example, so working with Laura Heiser, Lisa Cousins, and wonderful lab over at Oregon Health Science University, and Alana Ferdiga, who is now at Maryland, used to be at Johns Hopkins, and Dan Bergman and others, we asked them to help us come up with a tumor-immune interaction model, mostly focused on breast cancer. And very simple set of rules, right? In the tumor cells, oxygen is increasing cycle entry, pressure or compression is decreasing cycle entry. Oxygen, if you have low oxygen, you would go backwards along the curve and get more necrosis and vice versa. So, a handful of interactions like this. Starting off with naive macrophages that get activated into a pro-inflammatory state. And then if you have enough other contradictory signals, pro-inflammatory factors become anti-inflammatory. And then naive T cells can be activated to become effectors, but can also become exhausted. So just a handful of rules. So to a mathematician, it's like, oh my God, there's a lot of rules, how I'm going to code this thing. Well, now it's just writing out word sentences. We can write this model pretty much in real time. But let's see what it does. So let me pause this and go back. Here's just an example. Start off with the tumor. This tumor is kind of silly big, so it's become necrotic right away just by oxygen perfusion. And I'm going to start with a mix of unactivated macrophages and T cells. So here my necrotic cores emerge, which makes sense. Now the macrophages are going to be attracted towards that cell debris, start activating to become pro-inflammatory. That's going to start activating T cells to start nibbling away at the tumor, but eventually it's going to become anti-inflammatory. So you can see that you've got that early response in the tumor, but eventually we're going to go more towards an anti-inflammatory state, and that immune response is unfortunately very fleeting. But from a very simple set of rules, this cascade, right, from early growth, pro-inflammatory, anti-inflammatory, just emerges naturally from that set of very simple rules. So it gives us some kind of hope. Now, you may notice that I have JPEG compression. The other thing you may notice is that I wrote this thing in 2D because it's just way faster to iterate through a model and try 2D runs. So what about 3D? So I'm glad you asked that question. So you can take the same rules, and there's nothing implicitly 2D or 3D about this. The rules are just saying, if my cell encounters a signal, how does the behavior change? So 3D is just changing your initial behavior, your initial conditions and your geometry. The only rule we had to change to go from 2D to 3D was pressure, because it depends upon the normalization depends upon the number of neighbors, which would be 6 in 2D and 12 in 3D. That's the only rule I had to change in this simulation. And then after that, the 3D model comes for free from the 2D model. And you get the same kind of interesting things like immune cells coming together and forming a temporary aggregate until the chemical gradients disperse, and there's nothing to stabilize it right now, like say an APC, and they'll go off and attack and do interesting things. So you can go from 2D to 3D without much work here. So it's really, say, work where you're faster and then keep going. So Alana's group then at Maryland now has been really amazing at taking Visium, so spatial transcriptomics data, to say, now can we look through and use the gene sequencing to identify the cell types and identify their positions and nucleate as initial conditions a model. So patient specific, patient calibrated, no data simulation. So not a digital twin, let's be nice. But the neat thing is she had a hypothesis based upon earlier genomics work saying that contact between a regular epithelial cell and a cancer-associated fibroblast would tend to turn it more towards an invasive and EMT-like phenotype. And so the initial conditions and the arrangements of the fibroblasts and the epithelial cells would make a huge difference in the overall trajectory of this. And so that was kind of neat to say, I have identical model rules in these two, but just different initial conditions, and I get very different long-term outcomes here. And outcomes that are consistent with what we see clinically for this EMT. Another thing that Alana's lab then said is, let's build different models of immunotherapy by saying, different rules would say this immunotherapy does this or that. And we could also go to individual patients and say, look at the sampling of different immune types. And we would then randomly sample in the environment to place them, but according to the composition for the individual patients. So again, same rules for all the patients. So supposedly the same biology, just differences in the immune composition. And what you'll find then is simulating these different therapies, that the ordering of the response, let's say therapy 1 is better than therapy 2, better than therapy 3 for patient number 1. Go to patient number 2, that ranked ordering changes. And so the rank ordering of the responses really varies with those initial conditions quite a bit. So there's that really key important component. Not digital twin yet, missing data simulation, but patient calibrated, it gives us some hope. So we need some help to figure out how to bridge that gap to get to the data simulation. But it gives us some hope, right, that we can build a class of models expressive enough to capture many different kinds of trajectories. And given the right kinds of data, we can start to calibrate them, or at least initialize them to individual patients. And so if we can get the rules for patients, say by PubMed or by learning from the data, say from the transcriptomics, we could really build these things. And so then, she was working on these combination trials. So I should have shown this slide when I was talking about it. But the interesting thing, right, is that which therapy is best really varied by the patient basis. So you could do virtual trials with this kind of thing. And we would hope that you could start calibrating to individuals with the right techniques. Just as a proof of concept, you can actually dynamically add and remove rules in the middle of a simulation run. So I just made a quick code that says, for each cell type, pick a signal, pick a behavior, pick a direction randomly, and introduce a new rule dynamically in the middle of the run. And by howdy, the thing didn't segfault, it actually ran. So, there is some possibility that we might be able to kind of add mutations by adding and removing rules, or do, rather than parameter sensitivity analysis, we could do model structure sensitivity analysis. Say, do a random addition or removal of a rule, do we still get the same behavior and the overall model result, can we come up with, see how robust our model structure is? And that's something we've just barely begun to scratch the surface of, and we need help for that kind of thing. So the grammar, is in a sense agentic AI ready. So I guess let's do the tired and wired meme. Tired is you fire all your software developers and mathematicians and you vibe code your way to a model. It generates some code, it does something, it looks reasonable, it might be a hallucination. By the way, whoever invented hallucination instead of falsification or fabrication is a marketing genius for the field of AI. Side story. But the problem, right, is that you get this Gen AI curse. It might be wrong, it might not be wrong, probably it's wrong, but it looks awfully convincing and it's impossible to debug, and you can't reuse this thing. So I guess the wired is that you use AI not to write the raw code, but AI to write the model rules in the grammar. Something that we can interpret the output of, we can read this thing, we can combine model rules generated by AI with model rules made by humans, and combine these, they're fully compatible. And then that is an interface to well-debugged code that you don't let AI touch. And that's something that I think we could do. And then let AI work on the tail end. So down the road, you could say agentic AI says, I want to build a model with these things. It builds the rules, launches the model, and then it can interpret the results when you're done. So a model grammar becomes an interface to AI. So I think that's a possibility for building future work. So we really see multiple paths of combining human expertise, machine expertise. It's all mutually compatible, and we can create libraries of these things. And that's something I'd like to start doing in the future. So really, we want to say, can we do automated model generation and annotation? Can we learn models via large language models, for example? Also, we want to say, can we bundle up the rules for a different cell type and say that's like a cell definition or a cell line, and put it into a library that gets curated by the community, so more people can come along and say, here's a new model rule that worked for me, let's put it in and share this thing. And so then if you're reading your spatial transcriptomics, rather than starting de novo, you can say, oh, I've identified CD8 here, CD4 over here, here's a neutrophil, here's a Treg. Pull those types out of a library and initialize them in the right conditions, and we have a starting point. In fact, that's a grant that we're working on right now. So I want to thank really a huge network of wonderful people we work with, especially at Maryland and Johns Hopkins. The Galaxy team has been helping us build the cloud version of this, although today it doesn't seem to be working, so maybe I need to go talk to Enos and Jeremy, and want to thank the NIH for funding. And you can find this grammar was just published in Cell, which surprised the heck out of us. So you can download this paper and read about all the reference models and supplementary material. So please do read the supplementary material. And if you want to try learning to use the code, we have some training materials available. I think we have 4 minutes and 45 seconds left for questions. So thank you very much.
Thank you very much. So, yes, any questions?
A few, okay.
Trevor first and then Mark.
It was a beautiful talk, thank you.
Well, thank you.
So you can build different models very easily now, which is very impressive. When you're looking at the models, is there an easy way to tell if the model is not very good or if the parameters are not very good?
I think this is an area where now it took so long to get to the point where it was easy to build models that now we've opened up this class of questions saying, now I can build a model easily and iterate through, how do I know if it's right? And so we are doing a lot of work on saying, if you have quantities of interest, that defines an error metric, and can you automate minimization of the error metric to do model fitting? So that's something we're actually starting to build tools for now. And the interesting thing, though, right, is that there's quantitative matching, which we as mathematicians and models are more used to. But there are also qualitative predictions, right? To say, this tumor ought to grow and form a ring structure, or I ought to form invasive things, or I ought to look for the formation of little temporary immune cell aggregates. More qualitative statements, I think, almost are more image processing statements. And I think that's a whole class of other model validations that desperately need help from the community to build.
Mark, and then go to the back.
And please pardon me if I don't hear you very well. I've got robot ears.
Shout, Mark.
Please do.
No pressure.
Thanks very much, Bob.
Thanks, Mark.
How long will it take to get a virtual digital twin of a tumour then?
I'm sorry, how long is it what?
How long will it take to get a virtual tumour twin?
Well, depends what you mean by get a virtual. That coding now takes a few minutes.
Yeah.
Simulation run will take on the order of minutes to hours.
Okay,
to
I
do
mean,
a
how
3D
long-
tour.
How much more work needs to be done?
How
Will
much
we
what?
have what? How much more work?
Yes.
Oh, I see.
Will we have it
Oh,
for
okay, more
Christmas?
than that,
Will
no
it
plus.
be
Yeah,
in
5 to 10 years.
2030?
Oh, you know, about
Just put you on the
2092.
spot.
You know, I think, you know, now I think we have a lot of the tools where we've made it really easy to build models. We need people to know what they're doing to help us build those models. And we need other people who are good at the data assimilation tasks to say, can I actually assimilate in data and start making it look like a virtual patient? I think in this room, I think we have the pieces, and we could get somewhere. And rule discovery and all the things that go around it now are the things that we need to do as a community.
So back, Anna and Simon. I got it right.
Yeah.
Yeah, good. Yeah, sorry.
Thank you so much. My question is, you showed the transition between the 2D to a 3D model. How important is the assumption of sphericity in doing that? Because
So how do I look at the assumption of 3D versus 2D? So it turns out that for, you know, this is a question we asked for a long time in some sense. Before we ever got to the grammar, you know, we did, at first it was like, okay, we're gonna do everything in 3D. You know, Dr. Custom inside to beautiful 3D work, angiogenesis models in 3D. And what we found then is that we started getting to a point where like, you know, let's prototype models in 2D and then go back and verify if the same things pop out in 3D. Again, these qualitative predictions. We found for many classes of problems, at the very least we'll get the same qualitative behaviors. If the therapy works in 2D, it works in 3D, for example, right?
Are you making any
Yeah.
constraints about sort of
Yeah, I guess maybe I'm misunderstanding
radial?
the question.
Is that your question?
Yeah.
more so, is that similarity in qualitative behaviors dependent on an assumption of perfect sphericity?
Yeah, I think there are going to be some questions where I think 2D versus 3D matters. I mean, the canonical one is angiogenesis. In 2D, anastomosis is guaranteed because you're bound to run into a line when you're wandering. In 3D, anastomosis requires active guiding. It's about impossible to accidentally run into a line in 3D, right? So there are some problems where the topology of the problem is very important and to know in 2D versus 3D, right? It's like the old classical, you know, flatland, you know, where you have organisms that don't have a concept of 3D, and so like you get trapped in a house and die or something, or your digestive tract is open from front to back. Okay, so that's kind of scary. So if that thing moves into 3D, then it's like, oh, I spilled. So, you know, there are very important problems where that kind of topology really matters. I think if you get down to the fine-grained quantitative, it's also going to start mattering quite a bit, right? Because the number of paths you have to approach something in 2D versus 3D is very different, right? So often you'll find that's qualitatively correct, you know, and same, but say finding the bifurcation tipping point is going to make a big difference. So if that's what you're trying to predict, this precise tipping point, you're going to need the 3D. If you're saying, is there a tipping point, probably you can get that prediction from 2D. And so I think it really depends on what kind of question you're asking.
Okay, two very quick questions, Anna, then
Yeah.
Simon.
Thank you very much for an inspiring talk.
Thank you.
My question is really sort of looking ahead. I think somebody asked, when would you get a digital twin? Assuming we have a digital twin now, we will have another one in 10 years' time, in 20 years' time. How do you envision the regulatory support and, you know, all the ethics related to that? Maybe if you could comment on that topic.
Boy,
because when that comes in.
I mean, so, you know, and I think that there's going to be, you know, a classical, you know, like a digital twin is going to be really, I think, domain specific, right? Rather than, you know, people have this science fiction notion like going to make an entire digitalization of the entire body down to the, you know, the cell level and the protein level and simulate everything. You know, like, no. I think you're going to have digital twins of individual tumors and maybe, you know, some immune component, maybe a toxicity digital twin, and then you start combining these together into modules. And I think what I'd like to see, or hope to see, expect to see, is that people are going to develop components that are themselves working pretty well and then start combining them together into networks that make a better digital twin of more complete systems. So, you know, you have the tumor growth one, then you have the PKPD one, then I was really excited to see some of the talks this morning, right, about predicting the toxicity, right? So if you combine these different elements, I think you might be able to have something that you start supporting in the long term and then saying, can you build good enough interfaces that can start to network together? I think something like that. And they have to be versioned. I mean, we would hope that we can come up with better models over time, right? So you're going to have to do, just like any other code, you know, numbered releases with release notes and a DOI or whatever it is. Say, this is this version, and I record that provenance to say this model result came from this version of the code, so that you can then reproduce that later and have proper benchmarking for improvements. So it's kind of a wandering answer, but I mean, I think components will come together.
Final question quickly before tea, coffee,
This is a
whatever.
very open, non-quick question.
Okay, great. This is food for
There might be tea
thought.
time then, I don't know.
Okay, go for it.
I was picking up on the unpredictability of the models.
Mhm.
As you were saying that these things just diverge.
Yeah.
And whether, so that speaks to how usable and useful the models might be as digital twins. Because if you can only predict 7 days,
Yeah.
7 days ahead before it diverges. And I guess what is it that underpins that? Are we in the sort of world of chaos and sort of, you know, dynamical systems chaos? Or is it just more sort of stochastic, is what you're sort of...
I think in some sense, those are kind of two sides of the same coin. And I think biology solves this by swarming, right? If you look at, you know, when we have small numbers of immune cells, that stochasticity really is a big deal. But if you can get many, many immune cells, then it starts becoming much more deterministic. And I think, you know, what we need to do is, as we build these models, you know, one thing that we need to figure out too, right, is what are the rules of cells coming in and out of the domain? I mean, they're not closed systems, right? So swarming, you know, you'll look in a model, right, and say, wow, my T cell population went up. Oh, my growth rate must be like, oh, a 3-minute times, you know, cell cycle. That just means you have a bunch of cells coming in from outside, right? It's a non-closed system. So I think we need to study these problems and identify in advance the ones we think are going to be more fragile, less robust, more stochastic, and those are the ones we have to pay closer attention to, do more replicates, and really be honest about our cone of uncertainty on those predictions. And I think that's where the ones we're going to need a lot more data simulation, to bring in more data to narrow that cone of uncertainty or re-steer the model, right? And that's, in some sense, data simulation is basically a forcing term that says, I've got new data points, force my solution back towards them, so I can kind of get back into the middle of my cone of uncertainty and narrow it down a bit, right? So I think over time, we will be able to bring in more data to bring it. It's getting more and more miraculous over time. And I think the combination of that and pre-studying the models to understand where we're going to have the biggest trouble and being very honest about how uncertain we are about the results to be, I think, a really key thing. I think there will be a lot of problems where we will be able to predict very well. And I think we've seen the results this morning that show that, in particular, some of the predictions of chemotherapy response are going to be very, very robust. The ones that involve immune systems where the stochastic effects are there and we haven't gotten them to swarm, well, maybe that tells us we better get them to swarm, so it becomes more deterministic. So one avenue that we might investigate as communities say, how can we turn stochastic dominated models and systems more deterministic? What would we have to introduce to get it to act more like a deterministic system? And maybe that's introducing another cell type. Maybe that's influencing swarming of immune cells. Maybe it's something else. But maybe that's actually a design element that we haven't thought about as theoreticians and engineers and clinicians.
But it could be deterministic and chaotic. So
That's true, that's true. Which means that you just can't go very far in your predictions.
So I think this is a great time to stop and have a break.
I
So
appreciate
thank
that.
you, Paul, thank you, Yasmin, also really interesting talks. Thank you, Brill.